Good day to everyone. I will be providing a very short pre-lab discussion on our next experiment, which is specimen preparation, particularly, of course, the blood. Okay, so in specimen preparation, we will be preparing serum sample and plasma sample. So dalawa yung output natin dito. So you will be submitting to us, your laboratory instructors, one serum and one plasma. So again, let's have a quick review first on the difference between serum and plasma. Okay, so when we talk about um, serum, okay, this is obtained, or a specimen that is obtained by centrifugation of clotted blood. Okay, so kapag clotted blood, meaning we have used a blood tube or a blood collection tube that initiates clotting of blood, such as yung red top tube or the serum separator tube. Okay, so how about for plasma? Plasma is obtained again by centrifugation of anticoagulated blood. So in here, we have used a blood collection tube that has the presence of anticoagulants such as the purple top or the lavender top, which contains EDTA. Okay, so kung napapansin nyo class, no, inuulit-ulit ko kasi this is very important kasi once na nasa hima kayo, once na nasa blood bank kayo, you will be encountering these terms, serum and plasma. Okay, so balikan muna natin to, no. So in order for us to produce or to come up with a serum or plasma, we have to process first the sample. Okay, so after collection of the blood, you will submit the collected blood to be centrifuged. Okay, so as I know that you have already used, or you are already familiar with the use of a centrifuge sa AUBF. So nagsicentrifuge na kayo ng urine. Okay, so same principle lang din siya. Since centrifugation is the separation of cells from its liquid portion. So ibig sabihin, for example, sa blood, sa clotted blood, for example, no, we will be separating the RBC, okay, the red cells, from the liquid portion, which is the serum. Okay, so how are we going to do this? We will be centrifuging, okay, the specimen for 15 minutes at 750 to 1,000 gravity. So yung 750 to 1,000 gravity is yung speed of the centrifuge. Okay, and in other references such as Strassinger, okay, other references such as Strassinger, yung time of centrifugation and the speed is 850 to 1,000 gravity for 10 minutes. Okay, so again class, since not every centrifuge has the same amount of speed, has the same speed, and depende na rin sa laboratory protocol, so since the speed varies and also depends on the centrifuge that you are using and also based on different lab protocols, so pwede pa ring magbago yung 15 minutes, pwede pa ring magbago yung 10 minutes, pwede pa ring magbago yung speed. Okay? Pero technically, yung recommended is 15 minutes at 750 to 1,000 gravity, and in other references such as Strassinger, the recommended is 850 to 1,000 gravity for 10 minutes. Okay, so after centrifugation, na-separate na yung serum or plasma from the red cells. Okay, so ano next na gagawin natin? We will already aliquot the sample. So ano ibig sabihin ng aliquoting? When we say aliquoting, it means that we divide or separate the specimens into separate containers. For example, you have already obtained the serum sample after centrifugation. Okay? So, ang gagawin natin is using a pipette or a micropipette to get the serum. Okay? We will be getting the serum. Okay? We will be getting and divide the serum into separate container. So, meron tayong another container wherein doon natin ilalagay yung serum sample natin. And also, same lang din kay plasma. Okay, ibig sabihin, ihihiwalay na natin yung serum or yung plasma doon sa red cell by using another separate container. Okay? So ito, I have already discussed this one kanina. Okay? And then class, no, kung nakikita ninyo yung naka-asterisk, it is recommended that samples should be separated within 2 hours or less. Okay? Huwag nyo na siyang patagalin. Okay? Kasi, to prevent contamination of plasma and serum by cellular constituents, yun nga yung red cells, it is recommended that samples should be separated within 2 hours or less. Okay? So, class, kung mapapansin ninyo, no, during the experiment niyan, dalawa yung kukunin nating sample. Okay? One, to be clotted in order for us to obtain a serum, and then one from an anticoagulated blood. Okay? Wherein, ang mako-obtain natin doon is plasma. Okay? So, after collection, okay, the blood sample that is contained in an anticoagulated tube can already be centrifuged right away kasi hindi na natin siya pinapaklat. Okay? So, after collection from an anticoagulated tube, pwede na natin siyang i-centrifuge immediately. Whereas, kapag nasa red top tube or serum separator tube, kailangan munang mag-clot ni blood, like for example, at least 30 minutes, so kailangan muna natin siyang stand. Stand muna natin siya, okay, in an upright position, okay? Stand muna natin siya and paklat muna natin siya before centrifugation. Okay? Again, class, in the sample processing, it is recommended that samples be separated within 2 hours or less. Ang rationale doon is in order for us to prevent contamination of serum and plasma by cellular constituents. Huwag na natin siyang patagalin ng more than 2 hours. Kung pwede, pagka-collect, okay, kung plasma ang gagawin, i-centrifuge na siya agad, i-separate na natin siya agad. For clotted blood, i-clot na natin siya, and then pag nag-clot na siya, centrifuge right away. Okay, next one. So again, for sample processing, no, I have mentioned pa rin na sample should be separated within 2 hours or less. Okay, why? Because some laboratory results may be altered. Example of the lab results that might be altered are glucose and potassium levels and lactose dehydrogenase or LDH. Okay, so to prevent contamination of plasma nga, no, and serum by cellular constituents, kailangan ma-separate siya within 2 hours or less. Okay? So, if not centrifuged or aliquoted within the recommended time, the results might be altered and it might affect some of the laboratory results such as glucose, potassium levels, and lactose dehydrogenase. Like for example, si glucose. Okay? Si glucose is pwede siyang, ang glucose result ng patient kapag pinatagal siya ng more than 2 hours, okay, madi-decrease yung value niya. Kasi, because of the process of glycolysis. Kasi, di ba, if the RBC or the cellular constituents are left in contact with the serum or plasma, the glucose value of the patient will be decreased, okay, and also the potassium level magi-increase naman siya. Again, si glucose magde-decrease, si potassium level magi-increase. Okay, bakit? Because cells, okay, use glucose to keep themselves alive and nourished. That is the process of glycolysis. So again, ma-alter yung lab result. For example, si glucose, bababa yung value niya, okay, and yung potassium level is increased. Okay, kapag si LDH naman siya, magde-decrease naman yung level. Okay? So, anticoagulated samples can be centrifuged immediately after collection. So, pagka-collect using an anticoagulated tube, pwede na siyang i-centrifuge immediately. Okay? And then another, samples collected without an anticoagulant. Ito yung kinaklat natin, no? Must be fully clotted before centrifugation. Okay? Because kapag hindi fully clotted yung sample that is collected without anticoagulant, the fibrin strands can that are not completely clotted can interfere with the chemistry analyzer. Kasi for example, you will be using a chemistry analyzer. So ang ginagamit natin usually dyan is serum. Okay? Ngayon, kapag yung fibrin strands hindi siya fully clotted, pwede siyang magbara. Okay? Kasi nagsisip yung analyzer, sinisip na yung serum, and if meron siyang presence of fibrin strands, kasi nga hindi yan completely clotted, pwedeng mag-cause yan ng interference sa analyzer. Okay? And then, blood tubes must remain closed before, during, and after centrifugation to avoid contamination, accidental spills, and evaporation of specimen. Okay? So, dapat, um, we, uh, yung blood tubes, okay, especially during centrifugation, should be closed. Naka-close siya, hindi siya bubuksan, no? Kasi there might be the possibility of contamination, accidental spills, and also the specimen can evaporate. Okay, so dapat it should remain closed before, during, and after. Okay. And then, balikan ko, no, yung clotting, kapag kinaklat natin yung blood sample in a coagulated tube, yun yung serum separator, or yung nasa red top tube, for example, yung kinaklat natin. No? While clotting, the blood sample should be in an upright position and wag na siyang i-disturb doon hanggang sa mag-clot siya. Okay? So, kailangan natin dito ng test tube rack. So, dapat nakatayo siya, no? The blood sample should be in an upright position and wag natin siyang istorbohin. Okay? And then also, before aliquoting a sample, make sure to label the transfer tube and compare it with the label on the specimen tube used for collection. So, be aware of the pre-analytical considerations, okay? Do not mislabel the sample. So, okay, before you get the serum to put it in another container, okay, make sure, okay, to label first the transfer tube. Tapos, compare it with the label on the specimen tube that is used for collection. Dapat pareho siya. Okay? Kung dun sa pinagkuhanan nyo ng serum, ang nakalagay is 1 de la Cruz, dapat dun sa transfer tube nyo, okay, wherein andun si serum or wherein nandun si plasma, dapat same lang siya na may pangalan, 1 de la Cruz, and other details of that particular patient. Dapat balanse siya. Dapat pareho sila ng label. Do not forget that. Okay. What are the rules for centrifugation of samples? So I think your AUBF instructors, lab instructors, have already discussed the rules for the centrifugation of samples kasi pareho lang. Okay? So let's do a short rundown of this. Okay, yung mga important lang. Okay? The tubes that are placed in the centrifuge must be equally balanced. So, dapat balanse siya, pareho sila ng volume, pareho sila ng laki. Kung hindi, okay, kung hindi, pwedeng mag-break yung tube that is placed inside the centrifuge, and then also yung sample, matatapon. Okay? And also, magalaw yung centrifuge kapag hindi siya equally balanced. Ma'am, paano kung isa lang yung blood specimen? Ano yung pambabalance ko? Pwedeng lagyan ng water yung isang tube na pambalanse natin. Okay? We can use water, okay, na ilalagay natin dun sa tube na pambabalance natin, dun sa specimen natin. Okay? And then second one is to operate the centrifuge when the top has been firmly fastened down. Dapat nakasara siya. Papaikutin siya. Okay, before siya paikutin, dapat nakasara na yung centrifuge. And then also, while the rotor or while the centrifuge is still rotating, okay, wag nyo siyang open yung top. Hayaan natin siyang mag full stop. Okay. And then the third one, when a tube breaks in the centrifuge, stop and unplug the centrifuge, and broken glasses should be completely removed inside and should be cleaned and disinfected. So this is inevitable, class, kasi po pwede talagang magkaroon tayo ng breakages. Okay? But of course, kailangan once we have tubes that are broken inside, these broken glasses should be completely removed first, okay, and cleaned and disinfected before using it once again. Okay, so kung nakikita nyo yung image dyan, okay, yung naka-red na dyan is the tubes that has a blood sample. So 5 lang siya, di ba? So kulang siya, hindi siya balance, no? So ang pinang-balance dun sa isa is yung nasa yellow. So ang pwedeng lagay dyan is water para mag-balance siya. Okay? Again, class, make sure that the centrifuge is equally balanced. Okay? Meaning, you should use equal sizes of tube and volume across from each other. Hindi magkatabi, class, no? Dapat across. Because failure to do this, the centrifuge pwede siyang mag-vibrate and may possibly break the tubes. Okay, so class, no? Dito yung step-by-step procedure natin. Okay, step-by-step procedure, we will be collecting the sample. Okay, so we will be collecting two samples dito class. One to be clotted and one na anticoagulated. Okay, so once we have already obtained the sample, okay, yung anticoagulated blood can already be centrifuged immediately, while yung coagulated should be in an upright position, undisturbed, and let it clot for at least 30 minutes. Dapat fully clotted siya. And then after siya fully nag-clot, ikutin na ulit natin siya sa centrifuge. And then once na-separate na yung serum and plasma dun sa red cell, we will already aliquot the sample. Okay? And then, again, put the label before aliquoting. Before aliquoting. Okay, tandaan, before aliquoting, kailangan may label na yung transfer tube natin. Okay, and then you will be submitting two outputs sa lab instructors ninyo, one serum and one plasma, properly covered, okay, using a parafilm and properly labeled. Okay, so I think that is the end of our discussion. So I will be posting an assignment, okay, with regards to this discussion, and then next meeting, hopefully pag face to face na tayo, we will be doing this activity per group. Okay, so please check on the assignment tube in a while, okay, as I will be uploading a very short activity and assignment for you to accomplish for today. Okay, thank you so much.